So my name is Alon. Thank you very much for the organizing committee for inviting me today. So a little bit about me. I did my master's with Uri Gofna at Tel Aviv, and then a PhD with Dan Taufik at the Weizmann Institute. Then after 5 years in the Israeli biotech industry, I transitioned to a postdoc here at UC Irvine, where I'm using synthetic biology to study, to generate systems for rapid evolution of antibodies. So it's really hard to overstate the importance of antibodies in life science and biomedicine due to their exquisite capabilities to specifically and strongly bind to their targets. Antibodies have been considered the magic bullet in the treatment of various diseases such as infectious disease, transplant rejection, cancer, and autoimmune disorders. But still today, the vast majority of monoclonal antibodies are generated via traditional methods, which are animal immunization. And only 9 out of the 82 FDA-approved drugs were generated via synthetic or in vitro systems. And this is not surprising given the fact that when you introduce a foreign antigen into an animal, you exploit this very powerful response of the germinal center, where B cells are first selected for antigen binding, and then they undergo this very strong evolutionary process of affinity maturation to turn them into high affinity binders. But there are many issues associated with animal-derived antibodies. It's a very slow and expensive process. It's also an uncontrolled process. You can't select the epitope. It's inaccessible to self-antigens, to complex antigens like GPCRs. And there's also many ethical issues and reproducibility issues associated with animals. So synthetic or in vitro systems for antibody engineering do exist, and those are mainly yeast and phage surface display. And they've been the working horse in the field for the past 2 decades. The issue with these two systems is that you're ultimately limited to the initial complexity of the library that you introduce into the system. So the genes don't continue to diversify like in B cells. So you're very limited in your sequence space exploration. So an ideal system for antibody discovery and engineering would be based on an in vitro display technology, such as phage or yeast. But the antibody encoding gene would be continuously diversifying, like what's going on in B cells when they affinity mature. So for this purpose, we designed a system called AHEAD, which stands for Autonomous Hypermutation Yeast Surface Display. AHEAD is based on Saccharomyces cerevisiae yeast that harbor this very special plasmid called P1. So P1 is a linear plasmid capped by these 2 terminal proteins that's situated only in the cytoplasm of Saccharomyces cerevisiae. And you can clone your antibody gene or an antibody gene library onto P1, fused to the AGA2 cell wall anchoring subunit that allows it to be displayed on the cell surface. And P1 is being replicated by this special orthogonal DNA polymerase that replicates only it with a high error rate. So the error rate of P1 replication is 100,000-fold higher than the error rate inside the nucleus. And this defines a very streamlined process in which all you need to do to diversify your antibody gene is to grow the cells for a couple of days, and then the antibody gene gets diversified automatically. And then you mix up your yeast cells with some fluorescently labeled antigen, and then you can use FACS selection to enrich for cells with higher binding affinity towards your antigen. And as the cells recover from the FACS selection, they re-diversify the antibody, and that takes another couple of days. So then you can reiterate this process as many times as you like with very little work. All you have to do is just grow up the cells for a couple of days and fax them for like 20 or 30 minutes. So to test this system, we decided to use the SARS-CoV-2 trimeric spike protein as our antigen, more specifically the receptor binding domain of it. That's the part that latches onto your ACE2 receptor where you want to neutralize to prevent or to treat infection. And as a platform for antibodies, we decided to use nanobodies. So nanobodies are these small single domain variable fragments derived from camelids, camels, llamas, alpacas, and they're much smaller and they're much easier to engineer and manipulate. So this is how a typical affinity maturation campaign using AHEAD looks like. In this example, I'm showing a nanobody called RBD1 that binds RBD. So in the first round, you can see that there's very little binding, although we're using a high concentration of the antigen. But then within every cycle of AHEAD, there's an emergence of a new population. And again, this entire experiment took only 2 weeks with very little work. We just had to grow the cells and then fax them for 20 minutes. And because it was so easy, it was, we could scale up this experiment and I could Affinity mature 8 different nanobodies simultaneously, again, with very little work. So all these nanobodies were evolved for 6 AHEAD cycles with very, very little effort. And that resulted in high affinity binders. Here you can see a scheme of a 3D structure of a nanobody. And you can see that as expected, most of the mutations occurred in the complementary determining regions. Some mutations also occurred in the frameworks. We wanted to validate that our mutants actually increased in their affinity towards the antigen. So we ran a surface plasmon resonance study. So indeed, all of them increased. I'm just highlighting a few examples. So for instance, this nanobody RBD10 started off with a very low affinity, but ended up after 6 cycles with almost 3 orders of magnitude increase in affinity with a sub-nanomolar affinity. We also wanted to test the functionality of our nanobodies using a pseudovirus neutralization assay, like Ina just showed. And indeed, all our nanobodies were very functional, very potent. These are actually potencies are among the highest reported in the literature for SARS-CoV-2 neutralization. We also noticed something interesting that, for instance, this nanobody here, evolved nanobody, RBD11I14, which was our best binder, was actually a very modest neutralizer. While this nanobody here, RBD11I12, was a very modest binder, was our most potent neutralizer. And that led us to think that there's probably some functional diversity within their nanobodies, and they bind different epitopes on RBD2. So to investigate that, we devised this assay for ACE2 competition based on biolayer interferometry. So in this assay, it's pretty simple. We just use a streptavidin-coated sensor, and we flow the RBD, biotin-related RBD on it, so until we get saturation signal. And then we flow in the nanobody. So the nanobody binds RBD, again, until saturation of the signal. So you can see it here. And then we flow in ACE2. So if ACE2 and the nanobody do not compete over the same epitope, you'll get an increased signal. But if they do compete over the same epitope, you will not get an increased signal. These 2 antibodies are just known examples from the literature, from 3D structures where we know that they either compete or do not compete with ACE2. And indeed, we saw that strong binder weak neutralizer did not compete with ACE2, while the 2 other strong neutralizer either completely or somewhat competed with ACE2. We further wanted to look into what exactly is the epitope. We didn't do structural biology. Instead of that, we decided to do a deep mutational scanning library. So we borrowed this library from Jesse Bloom at the Fred Hutch in Seattle. So in this library, yeast are actually displaying the RBD. More specifically, a deep mutational scanning library of RBD, where each position in RBD is mutated to all the other 19 amino acids. And we actually run FACS selections against antibody binding. We're actually enriching for the escape fraction. Using deep sequencing, we get these logos that give you a rough estimation of what the epitope is. And we see that for that, again, for that RBD 10I14, the strong binder weak neutralizer, the interface, the epitope is actually distal from the interface between RBD and ACE2. While for the 2 others, the epitope is actually within, and that explains the neutralization efficiency of them. So just to summarize, and just one more point, we could see that we cover different mutations, and the functional diversity that we get within our nanobodies could be even served as some kind of cocktail to hedge against different mutations that occur within RBD. So because this mutation, for instance, E484, is a key mutation in for instance, in the Delta variant. Just to summarize, HEAD can be used for rapid affinity maturation of surface displayed antibodies. I showed nanobodies. We can also do this for SCFPs and FABs. Since it relies on simple cycles of culturing and selection, HEAD experiments are highly parallelizable and accessible, so everybody can use it. Parallel affinity maturation experiments increase the potential of discovering diverse sets of antibodies, so that's important to hedge against viral evolution, for instance. And I didn't show data about this, but library integrated into a HEAD can create a platform that mimics an animal's immune system, because you couple initial antibody discovery with affinity maturation. Just use the last minute of my talk. I want to tell you what I'm currently working on. So I want to turn a HEAD into a fully continuous system that doesn't rely on these discrete FACS selection steps. So in this case, the antibody and the antigen are displayed on opposite mating types of yeast. And since yeast must first bind each other using these surface proteins, affinity between the antibody and the antigen is directly coupled to propagation. And the way I do that is I use the proximity between the cells to signal using the mating pheromone, and I hijack the mating response. So instead of turning on the mating response, it will turn on a resistance marker or some kind of other growth marker that will be associated with propagation. And ultimately, the head cells will be competing with each other over faster propagation in a computer-controlled chemostat that require very minimal human intervention. And just want to thank my group members, and specifically Chang, my PI, and my collaborators from Harvard Medical School and from Duke University, and that's it.
Thank you, Alon, for a great talk. I'm going to start with a question myself, if you don't mind. So I think your future steps are probably going to answer that as well. But going back to the way that you produce these hypervariable nanobodies and the iteration that you go through, is it possible that during iteration you can also lose the affinity that you kind of select for? Because you start by sorting for something that binds very well, but then if they continue mutating with no selection step, then you can lose that affinity. Is that correct?
Well, that's actually true for any directed evolution experiment where you just like randomly mutate a gene. You always get stop codons, you get frame shifts, you can kill the protein altogether. So there's like a really fine line here of equilibrium between the mutation rates and the rate of selection. It was pretty easy to calibrate in our case because the mutation rate is not that high, actually. So it kind of worked out. You just need to grow the cells for 2 days and you always get beneficial mutations.
Okay, that's nice. Are there any other questions? I'm again suggesting the audience to write questions in the Q&A, and panelists, you can always raise your hand or directly ask a question. Well, I also see that we are.
I can jump in and ask one question. Yeah. So I was wondering if you could expand a little bit on the type of mutagenesis that this system enables. So is it sort of random mutagenesis, or is it directing it more down one route of like a certain type of changes that can be performed?
So right now the mutagenesis is based on orthogonal. It's an error-prone replication. Right. So this we engineered this polymerase to become error-prone. Right. The major problem with it is very transition biased. So we're kind of limited in terms of like which amino acids we can access from which, but it's based on replication. We just found out actually that there's like some kind of a gradient of mutagenesis along the P1 plasmid, but it's really uncontrolled. We're actually working now on targeting mutagenesis using other components like dead CRISPR-Cas9s. They're fused to 8 or other base editors.
Right, exactly. So that was my follow-up question, whether you can sort of target or rejig the system to find more mutagenesis space.
Yeah. Yeah, so we're trying to do that. Right now, it's completely random. So we're mutating the entire plasmid because it's based on replication.
Yeah.